Okay, guys, hello and welcome back and good morning. Today we're going to be doing the first lecture in the circulatory system, So, everybody heard of the cardiovascular system. That is not the circulatory system. They are not the same thing. They work together, but they are not necessarily the same. So, what is the, you like the backlights, not the front lights, the backlights. That's the backlights. That's it. Okay. All righty. So the cardiovascular system, cardiovascular. So this is a hybrid, two in one. Cardio means heart, vascular, blood vessels, blood vessels. So if you're just looking at a cadaver, a dead body, and you have the heart and you have the blood vessels, that is your cardiovascular system. What's missing? What's missing from this structure here? Heart, blood vessels. What else that could be a part of this heart, blood vessel anatomy and physiology? There's one more missing structure, and that's going to give us a whole third system. So those two, heart and blood vessel, that gives us the cardiovascular system. There's one more missing thing. If you add it to the heart and the blood vessels, you get the circulatory. You get the circulatory system. What could that be? What's flowing inside the heart and what's flowing inside the blood vessels? What is the heart pumping into the blood vessels? These blood vessels, these tubes, what is flowing inside them? So the heart is a pump, and the pump is pumping what inside the blood vessel? The blood. So the blood plus the heart and the blood vessel, that gives you the circulatory system. What are you circulating? You're circulating the blood throughout the tissue compartments of your different organ systems. Okay, so if you really want to understand the blood, what are we doing? We're doing anatomy and physiology, structure and function. What is the knife made up of? It's made up of a metal. Is it sharp or is it dull? So that's what we're going to be doing. So let's look at the structure and the function of the blood. So we're going to be looking at the anatomy, of the blood, or the structure of the blood. And we're going to be looking at the physiology. What's another word for physiology? Function. So let's look at the structure and function of the blood. How are we going to do that? How are we going to look at the structure and function of the blood? How about we centrifuge it? How about we centrifuge the blood? Obviously, if you look at the structure of the blood, what is that? Is it a gas? Is it a solid? Or is it a liquid? It's a liquid. So the blood is a fluid. What's a fluid? What's the difference between a fluid and a liquid? Are they the same thing? Of course not. Otherwise, why would they have two different names? In science, we don't have nicknames. So everybody gets one name that means one thing. So fluid and liquid, they're not the same. So what's the difference between a fluid and a liquid? Either we have a lecture in the heat and no noise, or we get to cool a little bit and it's really noisy outside. Well, what are we gonna go with? Leave it like that. Okay. So what's fluid? What's liquid?
Like between solid and gas.
So a fluid is both a liquid and the gas. So all gases are fluids, all liquids are fluids, but not all fluids are gases. So for example, when we do the respiratory system, air in the respiratory system, when we look at air in the respiratory system, that's a gas fluid. But when we look at the blood, that's an example of a liquid fluid. You see, air, taking a deep breath into the lung, exhale this carbon dioxide. That's an example of a gas fluid. Blood circulating in our veins and being pumped by the heart, that's an example of a liquid fluid. And we're going to be learning a very important concept in the dynamics of fluids. And that is fluids, whether gases or liquids, they will never move from point A to point B unless what? If a fluid is moving from point A to B, whether downhill or uphill, what does the law say? This is the biophysics. The biophysics of the fluid dynamics. What do we learn? We learn that a fluid, whether air, gas, or blood, liquid, will never move except from an area of a high, an area of a plus high pressure, of an area of a high pressure to an area of minus low pressure. If any fluid is moving, liquid fluid or gas fluid, it will never move except from an area of a high pressure to a low pressure. Who can give me an example of that? How about this syringe? If you have some liquid in the syringe, this is point A and this is point B. What's gonna lead the fluid to go from point A to point B? It's the pressure, because the pressure in point A is when you push with your index finger. So when the pressure in point A is so high, the pressure, because you're pushing the syringe, so the pressure in point A is so high, the fluid is gonna start to get displaced from point A to point B. And you will see that. Remember, what did we call the heart? A pump. So because the pressure is so high, when the heart contracts, the pressure is so high, the blood fluid starts to move from where? From the heart out into the system. Because if there was no pressure, had there not been any pressure, the blood would have never moved. That's why someone who is dead, why isn't the blood circulating in the body of somebody who died? Because their heart is not pumping, is not generating pressure. So the fluid is not moving. So if you get pushed on your shoulder, you tip over from the place where there's high pressure to a place where there's a low pressure. We will see the same concept with the respiratory system. Why did air move into my lungs? It went from the atmosphere, so that means the pressure was high out in the atmosphere, into my lung where pressure was low. That's when I'm inhaling, air went in. So the flow of the fluid, the flow of air fluid came from the outside atmosphere into my lungs. And when I'm exhaling, air is now flowing out from my lungs, out into the atmosphere. That means the pressure is so high. So my diaphragm moves up, compressing against my lungs, and the air is gonna flow out. This concept is very, very essential for you to understand that fluids, whether liquid fluid, my blood, or air fluid, the gas fluid, the air in my respiratory system, both share the same biophysics. They're not moving, no fluid is moving, whether liquid or gas, except what? Except there's pressure gradient. That means the initial pressure is so high compared to the final pressure. So you have a delta P, you have a gradient. You have a gradient where the final pressure is low, initial pressure is high, and then the direction of the flow of the fluid is gonna go from an area of a high pressure to an area of a low pressure. That concept is very important. So now back into the structure of our blood. How are we gonna figure out the structure of the blood? How about we get a test tube? We get a test tube. That's our test tube. And we fill the test tube with blood. Okay? Oops. We fill the test tube with blood. Then we put the test tube in. What are we gonna put the test tube in? What are we gonna put the test tube into? In the centrifuge. So we put the centrifuge. We put the test tube with a blood sample in a test tube and we spin it. Then after we centrifugate, we take out our test tube and we look at it. Is it gonna look the same way that it has entered? So what's gonna look different? What's your hypothesis? So it's very clear. You have a viscous blood sample, and you know blood has red blood cells, it has platelets, it has hormones, it has nutrients, it has toxic waste, it has different substance. What does the centrifuge do? Obviously, it spins. But when it spins, what happens? Yes.
Can separate the components of the blood.
Excellent. It separates the components of the blood based on color, based on size, based on mass. The answer is incomplete. It separates them based on what? Based on their look, based on their texture, based on their weight, based on what?
The quality of what's in it. I don't know. maybe.
Based on the weight. Good. You did good. You're doing fine. Excellent. So you got the part where it does separate. It fractionates. But it fractionates based on what? Based on texture, based on weight. So the heaviest is on the top or on the bottom? Excellent. So the heaviest parts are always going to be in the bottom, and the lightest part are always going to be at the top. Well, how many fractions are we gonna get? We're gonna get 3 fractions. Fraction number 1 is gonna be all the way in the bottom. Fraction number 2 is a little bit on top of it, and fraction number 3 is the topmost layer. This is really important. When you centrifugate your blood, you get 3 different fractions. In the bottom, you get about 45% of the blood. That's interesting. So 45% of the blood sample is going to be the bottom fraction. And 1% of the whole blood is going to be the middle fraction. And 55%, which is the remaining amount, is going to be the top layer. That's really important to know these percentages. For purposes of examination, you will be asked, after we centrifugate the blood, what's the map? How does the test tube look like? You say, we're gonna have 3 fractions, not 4, not 5. Those 3 fractions are based on weight. And because they're based on weight, they're gonna ask you, is the heaviest gonna be at the top or the bottom? You're gonna say the heaviest because gravity is gonna pull you down. So the heavier you are, the more on the bottom you're gonna end up. Then you're gonna be asked, what's the percentage? You will say the heaviest is 45% at the bottom, then there is a one layer that's about 1%. It is so thin, just 1%. And then the top layer is the most amount, that's 55%. Is that part clear? We're not done. Now let's describe these three layers. The bottommost layer is the layer that has the RBCs. What are RBCs? RBCs are red blood cells. So the red blood cells make up about 45% of your blood. Red blood cells have another name. And they are known as erythrocytes. Erythrocytes. So if you're told erythrocytes, you know it's the same thing as red blood cells. How much of your blood is erythrocytes and red blood cells? 45%. The top layer is known as the creamy or buffy coat. The creamy or the buffy coat. Creamy or buffy coat. What does it have? It has all the platelets and the WBCs. You see, we have RBCs and we have WBC. If RBCs, red blood cell, what do you think WBCs are? White blood cells. Excellent. So we have white blood cells and platelets. So you have to know that 1% is actually two different types of tissue of cells. Platelets are not cells, they are fragments of cells. But you have the white blood cells and the platelets. Platelets have another name. The other name for platelets is thrombocytes. Thrombocytes. Thrombocytes are your platelets. Thrombocytes are the platelets. So erythrocytes, red blood cells. Thrombocytes, platelets. What are white blood cells? Known as also leukocytes. Leukocytes. So white blood cells are known as leukocytes. So what makes up the 1% of my blood? It is the leukocytes and the thrombocytes known as the platelets. Excellent. Well, what's remaining now? What's the name of the top 55% now? That's your plasma. That is your plasma. Now, what is plasma? Plasma is water, H2O, plus nutrients. Everything that remains that is not thrombocyte, that is not leukocyte, and that is also not erythrocyte. So what else remains? Water, nutrients, gas is in there, oxygen dissolved in blood, plasma is in there, carbon dioxide, nitrogenous waste, metabolic waste, okay, hormones. So everything else is gonna be in that blood plasma, that 55%. So you need to know your three layers, and you need to know what is the name of each layer, who's the heaviest, who's in the middle, who's the topmost, the lightest. Each layer is made up of which type of cell. Your blood is very important. So that's the structure. So the structure, we already dealt with the structure. We have three different types of structures, 1, 2, and 3. The first structure is the red blood cells. Second structure is the thrombocytes and the leukocytes. And lastly, you have the plasma. Now let's look at the function of each. The function of the blood, obviously, since it has red blood cells are responsible for delivering oxygen to the tissue. and removing carbon dioxide from the tissue. So we will be learning that the red blood cells are constantly, they're like the subway. They have an uptown trip and a downtown trip. Each subway line has there's two different trips, uptown and downtown. So uptown is when we go from the tissue to the lung. And downtown is when we go from the lung back to the tissue. So if you're running the marathon, red blood cell is gonna run downtown from your lung carrying oxygen and giving your skeletal muscle. You're running the marathon, you need oxygen. I just came from up there, from the lung. So here you go, that's oxygen, that's a downtown trip. Oxygenating, unloading oxygen from the lung to the tissue. And then the skeletal muscles say, don't go back there empty. I have some metabolic waste. I have some carbon dioxide because I'm running the marathon. So in your uptown trip, go up and remove this carbon dioxide, dump it into the garbage, into the lung so we can dispose of it. So obviously my blood provides oxygen for respiration, aerobic respiration. That's why they call it aerobic respiration, because it involves oxygen, aerobic respiration. And it's also responsible for removing carbon dioxide, because carbon dioxide, if it's there, it's gonna increase acidity and cause a lot of problems. Also, the middle layer, thrombocytes. Thrombocytes, these platelets. What's the function of platelets? Platelets are band-aids. Platelets are band-aids. Why do we need band-aids? To stop the loss of blood. Why do people die from stabbing and gunshot wounds? Because they leak blood endlessly. And since we lose blood, we lose red blood cells, and the tissue is waiting for oxygen, but no oxygen is getting into the tissue, even though you're breathing, because you're losing red blood cells. Who's carrying the oxygen? The red blood cell, and they're being lost due to the gunshot wound and due to the stabbing wound, or maybe that's an example of an external hemorrhage. If somebody is getting stabbed or getting a gunshot wound, blood is flowing to the outside, that's external hemorrhage. Maybe somebody, you're looking at them, you don't see any blood leaking from their body. They have an internal hemorrhage, like a rupture of a blood vessel or an aneurysm, and the blood is leaking in the inside of the body. So it's still not getting to the tissue because it's hemorrhaging and spilling in the inside. So the blood platelets are band-aids that are gonna stick into the injury site, into the laceration, into the wound, into the abrasion, into the contusion, and prevent blood loss. We cannot afford to lose blood. Blood is so precious. You see what it has? It has so many things. It has plasma, it has thrombocytes, it has leukocytes, it has red blood cells. It is the most precious thing that you have. So we cannot afford to lose it. That's why we have the band-aids, that if you're injured and the blood is leaking out, we're gonna put a band-aid, a platelet. And also the blood is important. You can tell the function of the blood just by looking at the structure. You know, I just talked about the importance of blood in respiration, providing oxygen and removing carbon dioxide, because I saw who? Red blood cells. Thrombocytes, I know they're platelets, they prevent endless blood loss. Also looking at the leukocytes, white blood cells, these are the cops, these are the military. They fight the infection, the microbes, they fight the microbes. Also, you will see that these white blood cells are the neutrophils who target bacteria. You know how the Army has the Navy, the Marines, the different divisions? The same way, white blood cells, you have neutrophils that target bacteria, eosinophils that target viruses, basophils that target parasites, basophils that target viruses. So every different microbe, it has a special type of white blood cell that attacks it and defends you from it. Okay. So there were many myths that are involved with the blood. You know, some cultures, they assume that, you know, if you drink the blood of the lion, you become strong like the lion. Or if you drink the blood of the bull, you become like very strong like a bull. That is not true. And we discussed that was the cardiovascular system and the circulatory system. We said that the function of the circulatory system is to deliver oxygen to the tissue, remove carbon dioxide, and to provide nutrients. You know, when you eat a pancake, you know, pancake is carbohydrate. Carbohydrate is going to get the pancake will be digested in your digestive system. Then the digestive system is going to send it to your tissue in the form of glucose. Your cells do not see pancake. Your cells see a digested pancake in the form of glucose. Your cells do not see a burger. They see free fatty acid after it gets digested. So the food does not arrive to your cells the same way it was taken. It has to get digested first. And you know, if you take a digested carbohydrate meal of 5 separate people who ate different carbohydrate meals, you can never tell who ate what, because they get broken down into their building blocks, and the building blocks are the same. If you drink milk, that's going to be broken down into galactose and glucose, and galactose is later on going to be converted into glucose. If you eat oatmeal, that's going to be broken down into glucose and glucose. If you eat white sugar or table sugar, that will be broken down into that's sucrose, that will be broken down into fructose and glucose. Fructose is later going to be converted into glucose. So in the end, all the meals, the nutrition, the nutrients, how do they get to the tissue? In the blood. The blood is carrying red blood cells and the nutrients. Where do we find the nutrient? Do we find the nutrients in the buffy coat? Do we find the nutrients in the plasma? Or do we find the nutrients in the red blood cell? Red blood cell only carry oxygen and carbon dioxide in an uptown trip and a downtown trip. Where do we find nutrients? Remember, we said earlier, blood has 3 places. There's no 4th one. Either the bottom layer, which is all red blood cells, nothing else. Or the middle layer, which is buffy creamy coat, all platelets and leukocytes, white blood cells. Everything else is in the plasma. So where do we find the nutrients now? Plasma. Waste, where do we find waste? Plasma. Excellent. Everything. Hormones, where? Plasma. Stem cell, plasma. We also said blood is important for protection because the middle buffy layer, buffy coat has what? It has the middle layer has the white blood cells known as the leukocytes. Oops. Okay, blood is also important for fluid balance, stabilizing the pH and temperature control. How is that? You see, when you are hot, when it's really hot, what happens? Your blood vessels near the surface of your skin, they vasodilate. They become really wide. When the blood vessels become really wide, do they increase their amount of the blood that they contain, or do they now have less blood when they become wide? Less blood. So if I had a blood vessel this size, and it had this amount of blood, call it B1, then it engorged to this amount. Now it has B2 amount of blood. Who has more blood now? B1 or B2? B1 has more blood. Why do you think B1 has more blood?
We compare to the surface.
No. Okay, so they are the same. Maybe I'm not drawing it well. Okay, let me do this. They are the same length, the same length. Okay, the same length. Now the only difference is, so the length is the same. The only difference is the second one has more width. So now who has more blood? B2. So when you vasodilate, you accommodate more blood. So when you're so hot, your blood is going to become hot too. So the best thing to do, this is why when we feel hot, we go right next to the window. And you go, why do you go right next to the window? Because the window is cool, so the heat is going to flow out from an area of a high heat to an area of a low heat, right? From an area of a high heat to low heat. High pressure, low pressure. Same thing in your skin. Air is usually more cool than our body. Our body is really hot. So when you're running the marathon or you're running on the treadmill, you become really hot. Your temperature goes really high. So now all your blood is going to be shunted. All of your blood is going to be shunted to the surface. So now all of your blood vessels that are near the surface are going to vasodilate. So now there's so much blood flowing through your blood vessels that are near the surface of your skin. And now what's going to happen as a result? All of the blood is going to, all of the heat that's in the blood is going to dissipate. All of the heat in the blood is going to dissipate out to the environment and you're going to cool. That concept clear? That concept is really very important. So let's say that's your skin. That's your skin. That is your skin. If your temperature goes up, your blood vessels near the skin are going to become so wide and so much blood is going to be close to the skin. Now the heat is going to be flowing out from your body. This is your body and that's air. So all of the heat is going to flow out from the body into the air and you're going to cool down. The opposite is when it's freezing and snowing outside. If it's snowing outside, you're now cold. You don't have heat. You want to keep as much heat as you can. So the opposite happens. So the blood vessels that are near the skin, they become really tiny. And the blood vessels that are in the viscera, in the core of your body, they become really wide. And blood gets shunted now. It gets shunted into the core. So you want to keep it in the middle of your body so that the heat doesn't get lost. It makes no sense that it's snowing outside and the blood vessels near your skin are vasodilating. You're going to lose all the heat now. You're going to die from hypothermia. So that's how blood is important for temperature regulation. How is blood important for fluid balance? We talked about this in the gunshot wound and the hemorrhage, whether external hemorrhage, whether internal hemorrhage. If you are losing blood, you're losing fluid, you're losing water. This is why people get saline injection. Why do people get saline? They get saline because their blood pressure went down. So they get saline and the saline goes directly where? Into the plasma. So when the water in the saline bag goes into the plasma, what happens to the size of my blood? The size of my blood is now going to increase. It's going to increase. And that's going to restore my fluid. So we want euvolemia. We don't want hypovolemia. We don't want hypervolemia. We want euvolemia. Okay. What type of tissue is blood? You guys in AMP1 discussed the different types of tissues that are there in the body. The first type of tissue was what was the first type of tissue that you guys learned in AMP1? The skin. The skin tissue was made up of what kind of tissue? All that squamous and stratified and columnar. And cuboidal, what kind of tissue is that? I cannot hear you. Yeah, so that makes the skin. So part of my skin is stratified. Another type of my skin is mucosa. There's some of it is cuboidal. Others are columnar. Columnar what? Stratified means are they stacked on top of each other or not? Simple means they're only one layer. Cuboidal means they are equally wide as they are long. Columnar means they are more long than they are wide. But that's not the tissue. That's the description of the tissue. It's the squamous tissue. It's a columnar tissue. But what's the tissue that we are calling stratified? The tissue that we are calling columnar. What kind of tissue is that? Epithelial. That's the epithelial tissue. Nobody's going to describe nervous tissue as that nerve cell is a stratified nervous cell. No, we don't describe nerve cells. That's only unique for epithelial tissue. So the first type of tissue is epithelial tissue. That's the one we say squamous stratified epithelium. Squamous columnar epithelium. Simple cuboidal epithelium. So that's the first type of tissue. The second type of tissue is, good God, how long ago was AMP1? The second type of tissue after epithelial tissue is connective tissue. That's where blood falls. Because blood connect between the lung and the tissue, bringing oxygen, removing carbon dioxide, bringing nutrients, removing waste. That's a connective tissue. What other types of tissues are there besides epithelial and connective tissue? We have epithelial tissue, connective tissue. What else is there? We just did it. We just did it. The organs, the sense organs, input, receiving input, integration, generating output. What kind of tissue is that? Yeah, what kind of tissue that is able to receive stimulation, integrate it, process it, transduce it, generate an output to the effector? Can a connective tissue do that? No. Can epithelial tissue integrate anything? What kind of tissue is that? Nervous tissue. That's nervous tissue. And what's the last type of tissue that's out there? Muscle tissue. So these are the four types of tissue. So every tissue, histologically, we have to put it where it belongs. So the blood belongs to the connective tissue. Okay. There's a liquid connective tissue consisting of cells and extracellular matrix, the plasma, and it's a clear yellow, light yellow fluid. The plasma, do you think the plasma is as clear as water? Of course not, because water doesn't have electrolytes. Plasma does. Water doesn't have waste. Plasma does. Water doesn't have nutrients. The plasma does. So the plasma is a little more thick than water because it has proteins, it has electrolytes, it has nutrients, it has enzymes. But water is just H2O. What are the formed elements? The formed elements are the blood cells and the cell fragments. Who is the cell fragment? I mentioned earlier, somebody up there is not a cell. It's a fragment of a cell. What was that? Yeah, excellent. So the platelets are not cells. They are band-aids. They are fragments of bigger cells known as the megakaryocytes. So the formed elements, these are the blood cells and their cell fragments. So if you look down here, we have the formed elements, or some other books call them preformed, tomato. If you're reading the book and they say the blood has preformed elements, wait, but in lecture they say it's formed elements. Same thing, formed and preformed. What are the formed or preformed elements? The cells. My erythrocytes, red blood cells, my leukocytes, my white blood cells, and my platelets. These are what are platelets known as? Thrombocytes. There we go.
Erythrocytes are the, see where my mouse is? Erythrocytes are the red blood cells. Platelets are known as what? What are platelets known as? What's the name? So red blood cells are erythrocytes. Platelets are known as thrombocytes. And white blood cells are known as the leukocytes. More to know about the leukocytes is that some of them are granules, others are agranules. So when something says a granule, what does that mean? A granule means has no granule. Granulocyte means has granule. What's a granule? A granule is a big bag, a big bubble, big vesicle full of weapons. What are the weapons that the white blood cell need to fight the bacteria, the invading microbe? What weapons do we need? Acid, right? You need burning acid to burn the bacteria. So the granules are going to be full of acid. What else are the granules going to be full of? Enzymes, hydrolytic enzymes, degradative enzymes to chop up and destroy the membrane of the red bloods of the bacteria that we're trying to attack, of the microbes that we're trying to destroy. So you know the cops have to have weapon. It makes no sense the cop who's supposed to be fighting the criminals is just walking around with no weapons. That's the case in my country. So the cop is just walking around like that. So that's like a white blood cell with no granules. So all the white blood cells are fully, they have so many weapons: reactive oxidative species, free radicals, acid, hydrolytic enzymes, degradative enzymes, metalloproteinases, taser, gun, all of that. You need that. Okay, you're not going to be scaring bacteria and you have no weapons. But there are other types of white blood cells known as the agranulocytes. They have no bags. Does that mean they have no weapons? They have a different type of weapon that the granulocytes have, and that is, we will talk about that. That's the antibodies. Antibodies are not enzymes. Antibodies, they're like missile. So they throw, if the granulocytes did not kill the bacteria is too strong for the granulocytes, then the agranulocytes come in and help, and they have their antibodies. Okay, they have the cytotoxic T cells. We're gonna get to those. Just for now, know that your leukocytes, your white blood cells, they are there to fight the microbes. The neutrophils are there to fight the bacteria. That one is very high yield for purposes of examination. Which white blood cell is always gonna be targeting bacteria? So when you do blood work and you see the neutrophils are so high, you know what kind of infection do you have? You have a bacterial infection because the neutrophils are so high. So if you look here, so you see that's the neutrophil. You see the bag, you see the granules. The granules are inside this, the enzymes, the acid, the reactive oxidative species, the metalloproteinases, all of that is inside this bag. Basophils, you see their granules? They are full of bubbles, vesicles, granules. Red blood cells are not white blood cells. They don't fight anybody. They just transport, they are subway. They just transport oxygen to the tissue, from the lung to the tissue, and carbon dioxide from the tissue to the air. Platelets are band-aids. What's a monocyte? Monocytes are circulating immune cells that once they find the microbe, they differentiate into macrophages. You guys remember the Pokemons? You know, Pokemons evolve into something else. That's exactly what happens with the monocyte. So the monocyte is walking around as a monocyte. But once it sees a bacteria, once it sees a virus, it's going to evolve, we don't say the word evolve. This is not the Pokemon. We say differentiate. It's going to differentiate. is going to differentiate into what? Macrophage. So the monocyte is going to differentiate into macrophages. Are we clear? Macrophages. Are we clear? Lymphocytes. These are the B lymphocytes and the T lymphocytes. Remember, we discussed that when we were doing the endocrine system. Who grows in the thymus? Who matures in the thymus? T cells. That's why they call them T cells, because they mature in the thymus, beginning with T. B cells, where do they mature? In the bone marrow. So these are the lymphocytes. Oh, look at that. What is this? That is our blood. We took a blood sample. We centrifuged it. When we centrifuge this, you look now, we have 3 different structures. The topmost is the lightest. The one in the middle is the buffy, creamy coat, 1%, and 45% is the red blood cell. Red blood cells only, if you just take a sample of all the red blood cells, also called hematocrit. Hematocrit. So when you see the word hematocrit, don't be thrown off. It's the same thing as just all the content of the red blood cell taken out. So if we have the same blood, I can give you hematocrit, which is just my red blood cell, because you need red blood cells to survive. Okay? Plasma is going to be everything that is not thrombocytes and leukocytes. Is this figure clear? This is the most important figure of the whole lecture. Is that clear? Okay. Plasma is a liquid portion of the blood. And what is serum? Serum is the remaining fluid when blood clots and the solids are removed. Now, serum is very interesting because you know this blood is always rich in platelets in case that we are worried we will lose blood. We have platelets to prevent blood loss. Also, the blood has these coagulant substances. The coagulant substances make the blood really, really thick, and that prevents blood loss. You see, if the blood was thin, what we call them blood thinners, if the blood was thin, you're going to lose blood endlessly. That's why it's a protective, compensatory, adaptive mechanism to thicken the blood when you're bleeding. When you have a hemorrhage, when you have a gunshot wound, when you have a stab wound, God forbid, what happens is your blood naturally thickens up to prevent its loss. With the clotting factor, it gets clotted like honey. Your blood becomes so thick like honey. And that's to prevent its loss. So what is serum? Serum is the remaining fluid when you just remove all the blood clotting substances. So the serum is going to be just like plasma. So plasma has no white blood cells, no thrombocytes, no red blood cell, and it has all of the neurotransmitters and the hormones and the electrolytes and all of the substances. If you remove fibrinogen, what is that going to be? That's going to generate the serum. Now we need to discuss the plasma proteins. Plasma proteins, the most important and most abundant protein is the albumin. If they find that one in the urine, it means the patient has kidney failure, because that one is not supposed to get filtered in the kidneys, as we will learn when we do in the urinary system. Albumin is what keeps the water balanced, is what keeps the osmolarity and prevents any disturbance in the water balance between blood and the tissue. So there are diseases where you see the patients, their legs are so swollen with water.
Some other patients, that's known as lower limb edema. Lower limb edema is when there's so much water, the legs are so swollen. If you touch their legs with your thumb, a pit will form. They even call it pitting edema. Why? Because there's so much water in the tissue. There's an imbalance. It's what's supposed to be equal amount of water in the blood to equal amount of water in the tissue. But because of albumin imbalance, water is gonna flow into the tissue. Their legs are gonna be really, really engorged and swelling. Also, if you look at a disease known as Kwashiorkor disease, ever heard of that? Kwashiorkor disease. In Kwashiorkor disease, it's a disease of poverty and malnutrition. So there's no food in the body, no proteins. So the blood now really has no substance, no osmol, no solutes. So the tissue has more solute than the blood. So water goes obviously from an area of a low solute concentration to an area of a high solute concentration, and that's what causes that's what's gonna cause the swelling. So they can have really big belly, these people who are suffering from the Kwashiorkor disease. Globulins. These are proteins. So remember, albumin is important, as we said, for the osmolarity and the viscosity of the blood. Globulins are important to make the antibodies. So the antibodies are known as immunoglobulin. We will do that when we do the immune system. They're known as immunoglobulins. And fibrinogen, every time you see something ending with nogen, pepsinogen, fibrinogen, angiotensinogen, if it ends with inogen, it means that it's off, it's inactive. When it becomes activated, the nogen will get removed and you will have fibrin. So fibrinogen is inactive, fibrin is active. Pepsinogen is inactive, pepsin is active. Trypsinogen is inactive, trypsin is active. Angiotensinogen is inactive, angiotensin is active. So fibrinogen is a precursor that is inactive. But once it gets activated, you have fibrin. What is the fibrin going to do? The fibrin is like a meshwork that's going to mesh together like a net and bring all the red blood cells together. What do you think that's going to do to the viscosity of the blood? If you're going to mesh the red blood cells together, is that going to make the blood thinner or thicker? It's going to be very thick. So fibrin is important for the precursor for fibrin threads that help form blood clots. It helps forming the blood clots. Plasma proteins are formed by the liver, except globulins. Globulins are produced by the plasma cells. So people who have alcoholic liver disease, people that have cirrhotic liver disease, they have a problem making the plasma proteins. It means that they will not have what? They will not have albumin. You see these plasma proteins, albumin, fibrinogen, globulin, they will not have those, except globulin actually, because globulin is not made in the liver. Where is globulin made? Plasma cells. You see? Plasma proteins are all formed by who? The liver, except globulins. So people who have a liver disease, their immunity will not be affected because the immunoglobulins that we need in our immune system are not made by the liver. They're made by the plasma cells in the bone tissue. But the albumins and the fibrin protein are made up by who? Who makes them? Who makes these proteins? The albumin and fibrinogen. The liver. Excellent, the liver. Nitrogenous waste. So the blood is not only carrying good substance, it's also removing bad toxic waste, which is a good thing. So we're not only concerned with delivering good supply to the tissue, we're also concerned with removing all the harmful waste
from the tissue. Nutrients or glucose, and that's for the glucose metabolism. To get ATP, we need glucose. You eat pancakes, that's sugar. You drink milk, that's sugar. Sugar cane, oatmeal. So all of that is going to be broken down into sugar so I can have ATP. Fats, eating all the steak and all of the fatty food, you get ATP from that also. Proteins, that's an example of a nutrition also that we get ATP from. Vitamins, phospholipids. Phospholipids are used for the integrity of our plasma membrane. You replenish your own plasma membrane phospholipids. The blood plasma also has dissolved oxygen, so not all the oxygen is carried by red blood cell. So don't assume that 100% of the oxygen is here in the red blood cell compartment. Most of it, yeah, most of the oxygen, most of the carbon dioxide is found here within the red blood cell. But that doesn't mean there is no oxygen dissolved in plasma. There is some oxygen that is dissolved in plasma and some carbon dioxide that is dissolved in plasma also. But it's not the most amount. The most amount is in the red blood cell, not just the plasma. Electrolytes, a lot of sodium, potassium. Why do we call them electrolytes? Because they conduct electricity. If you have distilled water, what is distilled water? Distilled water is H2O only. It has no salt in it. It has nothing in it. So if you put an electric wire in distilled water, and then you dip your finger in there, are you going to get electrocuted? No, because electricity does not flow in pure water. But let's say you drop like a little bit of salt in there, and then you drop a little bit of salt in the distilled water, then you put electric wire, and then you dip your finger. Oh, you're getting electrocuted. Why? Because the water is going to cleave sodium chloride into sodium, Na into sodium, and chloride minus. So when you have the charge separation, plus minus, sodium is positive, potassium is positive, calcium is positive, chloride is negative. So now you see these charged substances. These electrolytes are important for our body for the action potential. Remember the action potential? So the sodium influx causes depolarization, potassium efflux causes repolarization. So all of that is carried in the plasma. Remember we discussed in homeostasis, we don't want hypercalcemia, we don't want hypocalcemia, we don't want hypermagnesemia, we don't want hypomagnesemia, we want eukalcemia, euphosphatemia, eumagnesemia, everything U. Is that clear? The viscosity of the blood, that is the resistance of a fluid to flow. The osmolarity, we discussed that. There we go, that's Kwashiorkor's disease. Remember we discussed that. What causes Kwashiorkor's disease? Why these kids have, if you see these kids have really big bellies, or you see an elderly person with really huge swelling feet, and you put your thumb in there and it makes a bump. What kind of edema do we say that was? Who caught that one? When you dip your thumb into the swollen legs of a patient who has lower limb edema and you press against it, it makes a little depression. What is that called? Pitting edema, because you make a pit. So that is pitting edema. Let me see if I can find an example. Do you see pitting edema? Okay. Can everybody see pitting edema? So what happened to this patient? This pitting edema is caused by severe excessive fluid accumulation. That's just like the babies who have really big bellies. So what happened? What happened was, in our body, we have two compartments that have to be in balance. So the scale has to be balanced. The scale has to be balanced. Equal. There has to be equality between equality in what? In the solute or the osmolarity. Osmolarity. Solute osmolarity. There has to be an equality of solute osmolarity between two compartments. Number one, the blood. Number two is my tissues. You cannot have the blood hyperosmolar to the tissue. You can have the tissue hyperosmolar to the blood. They both have to be equal. So how does edema happen? Edema happens when water moves from blood to tissue or from tissue to blood. Yeah, from blood to tissue, when water moves from blood to tissue. This patient has more water in their leg skeletal muscle tissue than in the blood. What did you learn the first thing when you took A&P1 about osmosis? Osmosis says that water is gonna flow from an area of a
Where water is.
from an area of a low concentration to a high concentration. If you have 25 millimolars of sodium chloride and 250 millimolars of sodium chloride, water is moving from low to high or high to low? From low to high. If you have 2,500 millimoles of sodium chloride, water is going from low to 250 or low to 2,500. To the 2,500. Water is always going to the area of a high concentration. So why did these babies have big bellies here? What's happening here is because they are so impoverished. They are so starved. They're not eating. Their blood doesn't have any protein. This is called hypoproteinemia. Hypoproteinemia. The blood has no protein, but the tissue has a lot of protein. So water starts moving from blood to the tissue in their abdomen. That's why they have these really big bellies. That make sense? Because of the hypoproteinemia. How is blood produced? Blood produced by stem cells. We will see in a figure that in your bone marrow, all the blood cells are made in the bone marrow. So there is a multipotent, it has multiple potential. One cell, it has the potential to become red blood cell. It has the potential to become white blood cell. It has the potential to become a lymphocyte based on the cytokine. Ever heard of cytokines? Cytokines. So cytokines are the signals that tell the stem cell, we need you to become a red blood cell. It now will become a red blood cell. Stem cell, that's a cytokine to make you a B lymphocyte. Now we will become a B lymphocyte. Stem cell, that's a cytokine that's gonna make you a neutrophil. Then it's gonna become a neutrophil. I think the figure should be, there we go. This is the figure. Where is the figure? That's the figure. You see, that's the pluripotent stem cell. There's a cytokine here in this arrow that made the pluripotent stem cell become a colony forming unit. Then another cytokine made the erythrocyte colony forming unit convert into a erythroblast. Then another cytokine makes the erythroblast become reticulocyte. Then another cytokine makes the reticulocyte an erythrocyte. So we begin as a pluripotent stem cell, then we end up with a erythrocyte. Does that make sense? There we go. That's the figure I was looking for. You see, that's the pluripotent stem cell. See where my mouse is? It's a one cell, but it can become this, that, or those two, or this, based on what? Please look at this very far diagram. If you see here, what do you see? Let's say a xenophil, that's a basophil, that's a neutrophil, that's a monocyte, that's a B lymphocyte, that's a T lymphocyte, and that is a natural killer cell. So based on demand, what is the body demanding? What does the body want? Does the body want a xenophil? Does the body want a basophil? Does the body want a neutrophil? Then it will go all the way back to the stem cell and tell the stem cell, we need a eosinophil. So the stem cell is now gonna go into becoming a xenophilic colony forming unit. Then it's gonna become a eosinophil. If we need a basophil, the pluripotent stem cell is gonna differentiate into a basophil. If we need to become a neutrophil, the pluripotent stem cell is going to differentiate into a neutrophil. Questions? Questions? No questions. Okay, so let's stop here and I'll see you in the lab up there at